Hello, everyone. I'm Christy Risk, Senior Editor at Genome Web, and I'll be your moderator for today. The title of today's webinar is How Low Can You Go? Reduce Breeding Costs with an Optimized Genotyping and Analysis Strategy, and our sponsor is NRGene. Our panelists today are Dr. David Heighton, Associate Professor and Associate Department Head in the Department of Agronomy and Horticulture at the University of Nebraska-Lincoln, Dr. Benjamin Kaufman, Director of Operations at Agriplex Genomics, and Dr. Edward Brugman, Director of Genomic Solutions at NRGene. You may type in a question at any time during the webinar. You can do this to the Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Heighton. Please go ahead.
All right, well, thank you very much. And I want to thank everyone for joining us today for this webinar. And I want to thank Agriplex and NRGene for inviting me to start us off today on today's webinar. So I want to start off today talking about how molecular biology enables real-world crop improvement. And part of that is really deploying genotyping and breeding and enabling genomic selection. And so the question, or part of the title for our talk today was, how low can you go? And with that question, we often think of that question of how low can you go in terms of cost, but then we also think of that as in terms of how to reduce that cost, but we reduce that marker numbers. But while we reduce that cost, we not only have to maintain our throughput, or in fact increase our throughput, but we also have to maintain qualities if we're really going to enable genomic selection, and if we're really going to deploy genotyping and breeding. But before we get into this, the cost and how low can we go, I want to talk a little bit about real-world crop improvement. And I'm going to use soybean as an example because that's the main crop that I work in. If we look at the USA on-farm soybean yield over the last 30 years, what we see here is that if you look at the trends of the on-farm soybean yield, it's gone a continual increase from 1990 all the way to 2020. And of course, there's peaks and there's valleys. You've got your good years, your good yielding years, and then you've got your low yielding years, good weather, bad weather. But if you draw a trend line through that, you can see an average rate of genetic gain. It's approximately about a half a bushel per acre per year, or about 33.6 kilograms hectares per year. Now, of course, research is the main the main bulk of this gain. It's what really helps us get this gain of yield. Now, you've got breeding and genetics is a portion of this, but you also got better farm management practices. That is the other portion. So the question that we have behind this, how much of this is breeding and genetics? Well, a group out of U of I did a decade study. Brian Deers led this study, and what they did was they took cultivars that were grown from the 20s all the way to current time, and they took them from different decades, and they grown them under the same farm management practices. And with this, they wanted to answer that question. Well, how much of that genetic gain that we see on these on-farm yield gains, how much of that is new genetics? And what they see is that historical gain in at least maturity groups 2 and 4 in soybeans was 20 to 23 kilograms per hectare per year, with the current gain rate about 29 kilograms per hectare. And so whenever you look at that on-farm rate of gain of 33.6, you can see breeding and genetics. If you're really wanting to affect real-world crop improvement, breeding and genetics is almost 2/3 of that gain, as a significant impact of what farmers are actually seeing as far as improvement in their crops. So when we look at the process, the breeding process, and I say this as a molecular biologist, and I see what breeders do. You take two parents, and you cross those parents, and then you got this black box of what breeders do. And, I look at them, and they grow some random plants out in the field, and they walk around in the field on nice days, and I don't know, they complain about the weather, and then they pick some plants that look nice. And once they pick those plants, voila, they've got a new elite cultivar. And then that's it. We don't really need to talk about anything else. Well, luckily the breeder next to me that has an office next to me is working from home, so he's not busting down my door. But that's not really, obviously, that's not what they do. There's actually a lot of science that goes behind breeding. And whenever we look at the real breeding process in soybean, I took an example from the soybean breeder here at UNL. On average, he's taking about 250 crosses that he's making between different parents, and these are, to increase not only yield, but also better disease resistance, better drought tolerance, whatever the traits he's targeting for increasing rate of genetic gain here in Nebraska. And after, F2 plants, there's about, he's growing, it's around 100,000 to 200,000 F2 plants in winter nursery, doing a single seed descent to F4 single plants. There's about 35,000 F4 single plants. And from these 35,000 plants, selecting about 20,000 lines, growing these as single row observations. Now, these F4-5 lines, these single row observations, they don't have that observation is not any real data. It's basically a visual look at that line from these crosses. But you can see from that 20,000, you're going from 20,000 down to 3,000. And if we look at the genetic gain over time, I put rate of genetic gain, and so I put that USA on-farm soybean yield on the bottom there, and that trend line, that rate of gain. If you look at the breeder's equation, that RT, that rate of gain is I, the selection intensity. How hard are you selecting through this breeding process? With that 20,000 down to 3,000, that's almost a 10% selection intensity. How accurate can you select it, that R? Well, a single row observation is not accurately selecting for anything at all. How much genetic variance do you have? Additive variance there. That single 20,000 gives you a lot of genetic variance. But as you go through that breeding process, 3,000 lines, 500 lines, 50 to 60, 20 to 30 lines, where if you look at that regional testing later in that pipeline where you have 15 plus locations of data, 2 to 3 reps, where you're actually getting good yield data, you've got very little genetic variance to select upon. 20 to 30 lines is not a lot of genetic variance there. And then how quickly can you cycle these lines back as parents to get cycles? And then part of that breeder's equation I have there, $sign, that's not really part of the breeder's equation, but I like to put that there because this is all in the context of money. If a breeder could afford to grow more than 50 to 60 lines across 15 locations, the breeder would. And that would add more genetic variance, which would help the rate of genetic gain. Well, whenever we try to integrate molecular biology into this process, you can really look at it across any generation here. You can integrate molecular biology or markers into this. Whenever it started out, I keep wanting to click my little arrow here. Let's see here. Marker-assisted selection, we started integrating markers at marker-assisted selection. And while you could do it at the F4-5 lines, in soybeans, F2 plants is a really great way to enrich populations. You can do 1 or 2 markers for key high-value traits that are large effect genes. Now, when we think about genomic selection, depending on how much that marker technology to run genome-wide markers cost, you might be able to integrate it at this F4-6 lines where you got 3,000 lines. But the cheaper that you can get, again, that how low can you make it go? How far early in this process can you get it? Those 20,000 lines where you got single row observations, you've got no phenotype data to make a selection upon. So you're essentially selecting randomly or visually. Genomic selection, as long as it's better than random, you're adding a selection accuracy there. You're increasing your genetic gain over time. And if you can even get it cheap enough, can you even push it back to the single plants before you go to that single row observation? So there's a lot of opportunity there. Other areas where you can integrate markers, you've got skim sequencing with imputation, where now in soybeans, we're able to do a 0.3x sequencing, impute up to 95% accuracy. 50 to 60 lines, we can do that from $30 to $50 a line. You could do this in this stage at 50 to 60 lines. Look at your haplotype variation in these more advanced lines. These lines could be used for your training population, so you have that high marker density in there. You could even potentially push it, as we get it cheaper, push it down to these 500 lines segment in this breeding process. And then as you're pushing these lines, more advanced lines, and so that especially as they become parents again, you don't want to be imputing upon generations. You're going to be doing whole genome resequencing to look at indels and then also correcting for that imputation so that you're not imputing upon each other. But overall, you can essentially integrate markers at different stages upon your breeding process. And also, just so the F1 stage is not left out, you can run markers at the F1 confirmation stage here too. But overall, the big picture here is as we go through this breeding pipeline, breeders are decreasing the number of lines they go through this breeding process, but they're increasing the amount of information they get on these lines, both phenotypically and also genotypically. And this really accounts for an increased investment on these lines so that the breeder can make informed decisions upon selection. So whenever we look at genomic selection, and that's what I really want to talk about, and it might be a little hard to see on the screen here, but that genomic selection, where we can push those 3,000 lines, 20,000 lines, from that breeding process, these are this biparental cross, lots of lines, but we want to get it cheap. And if we look at genomic selection, our training population where we're doing the skim sequencing with imputation, we're going to genotype those lines with a sequencer there and phenotype them in our target environment. And then we're going to plug those in, the genomic selection people, they plug them into their fancy and complicated models. I'm not a genomic selection person, so the best I do is I see, I can put that model training up there. But they train their model. We genotype the soybeans from the breeding process and sequence them and put them into the model. And then we predict and select the lines out of that. Now, the good thing is the soybeans from that breeding process, they're from biparental crosses, and they have very high linkage to equilibrium. And to show that, I took a line out of here, and this is very typical from a biparental cross. The parents, like I mentioned, the parents can have very high marker density. We can use that skim sequence with imputation and have them resequence. The RILs, this is a line here where I've coded one parent inheritance with the blue, the other parent inheritance with the red, and I'm only showing the polymorphic markers. I eliminated all monomorphic markers. This line was run with a 6K Infinium chip. And first thing, soy has very little diversity in elite breeding lines. And the first thing that really stands out is there's not a lot of diversity segregating in this particular line that you need to select upon. And you see here in RILs, you have 1 to 2 recombinations per chromosome. And so you don't need a lot of markers to select upon. And there's like 50 to 60 small diversity blocks that are segregating in this cross. So between the small diversity blocks, 1 to 2 recombinations per chromosome, the number of markers needed for genomic selection is really in the hundreds. You don't need high density data for these populations that you're selected upon. And so whenever I show that for multiple populations, I just kind of screen grab some multiple lines from different populations. It's not that different. You see, if anything, you see a lot of white space, but you can be very effective with a couple, 200 to 300 markers pretty easily. So when we wanted to enable genomic selection in soybean, we really thought about low density genotyping to enable it in this generation of these F4-6 lines, F4-5 lines, or even, ideally F4 single plants before we're even driving lines. And this is, looking at targeting sequencing where we could get it cheaply and really with a targeting price of $3 to $5 a sample. The cheaper we can get it, the earlier we can drive it in this breeding process. We need to make it high throughput. We're talking about tens of thousands of lines just for one breeding program. And we need flexible marker numbers so that we can get it a few hundred markers in a population. And it needs to be high enough quality so that it's effective for genomic selection. So the technology that we decided to explore in my lab was molecular inversion probes, or Mips. And one of the reasons why it's been developed for multiple species, it first was developed in human species, flexible marker targets, it can be 100 markers up to 50,000 markers, and it targets specific SNPs. So you can actually select the SNPs, you're not just getting random SNPs. In the literature, it had low missing data rate, so your data that you get back look more like array data than it does the restriction enzyme type GBS data that you see. And it's got high accuracy, and it can call heterozygous alleles. And so on the right side of the slide there, you can see the top line there has a linker. That green is a universal backbone. That's, if you have 1,000 MIP probes, all the MIP probes will have that same universal linker. Then the ligation extension arm is specific for each different SNP that you're targeting. And the target there on the next line, the B line, It can be 100 base pairs, 200 base pairs. We generally target it to be about 100 base pairs. The extension and ligation arm will flank it. You'll do an extension ligation step to create a circular DNA. You then do a universal PCR using a primer 1, primer 2 that's shown there. And that universal PCR will then tag it with P7, P5, which are the Illumina tags, which allows you to do Illumina sequencing. And at the same time, you can index it with I5, I7 indexes so that you can barcode it and multiplex samples. So for developing SNPs, we've really done 2 stages here. We optimized it for soybeans. We looked at different types of probe synthesis, pooled versus individual, DNA extraction, reducing that cost, trying to get down to that $5 a sample and high throughput. I'm not going to talk about that first part today. Just to summarize that, we settled on individual synthesis for the probes. It gave us a lot more flexibility and much more consistent for genomic selection. DNA extraction, we've got a method that works, but we're still working on improving that because I think that's still our key bottleneck. And reducing that cost, we've got it under the $5 a sample, so we're pretty happy where we're at there. And throughput, we're happy with our throughput too. I'll mainly focus most of the rest of my slides on this developing it for the genomic selection for soybean, where we selected out a 1K genomic selection probe set, and that we can use across multiple breeding programs, and the validation of that probe set that we're using. Our sequencing is on a NextSeq 500. We're using a custom primers because the MIP backbone, we needed to use custom sequencing primers. We do dual barcoding because we use a mid-output kit, and with that mid-output kit, that allows us to multiplex about 1,300 samples. So that is one of probably the biggest downside of this technology for us, is it requires us to be able to get runs of 1,300 samples. We just can't run 100 samples at a time. And so it requires us to coordinate lots of breeders at a time, or we have to make sure that we're coordinating samples in so that we get 1,300 samples ran at a time in blocks. So that is probably the biggest limitations of scheduling that we've come across so far. So in designing the 1K probe set, I've worked with Aaron Lorenz at University of Minnesota, and Ben Campbell was his postdoc or PhD student. I don't remember which one he was at the time. I think PhD student. And the SNPs were selected from the 6K Infinium chip that Kijun Song at the USDA in Beltsville had designed. And the 6K had been ran on regional uniform test lines that had been selected from North Central elite lines that is in that regional testing. And what Ben did was he divided the genome into 500 5-centimorgan bins. He called it this bin pick method. And he selected 2 SNPs per bin so that they'd have the highest polymorphic information content in the what he called the Big 9. And the Big 9 were basically lines that came from these programs because we were developing this genomic selection probe set to work in these different breeding programs. And what he found was using this method, and he compared it to other methods for selecting SNPs, and found that on average, whenever you took any random 2 lines from these different programs, it had on average 500 of the SNPs polymorphic compared to 427. Now, for genomic selection, we're going to be selecting we're going to be running the genomic selection within populations within the breeding programs, not necessarily across the breeding programs. So he did that same comparison within breeding programs. So this is those same the uniform lines, but broken out by the different breeding programs. So Minnesota, Illinois, Nebraska, and also there's some southern lines, so Southern Illinois, Tennessee. And so you can see for where this genomic selection probe set is designed for, Minnesota, Illinois, that bin pick that I've circled there, 443 on average SNPs. If you take any 2 lines within the Minnesota program, on average, you have 443 polymorphic markers versus randomly, you would only have 322 SNPs polymorphic. HaploView, we used it had 323, 332. Song method that he used for selecting the 6K chip, it was very comparable. a lot of the same, close to the same number of SNPs polymorphic as the bin-pick method. As you get to some southern germplasm, which this GS probe set wasn't necessarily designed for, you do get fewer SNPs polymorphic, but still a pretty good number. You see Tennessee there, 352. Now, while the number of SNPs polymorphic for the bin-pick method is very close to what the song method was, the advantage of the bin-pick method shows up whenever we compare it to the distribution of the markers. So what I've showed here is the song pick method on top of 7 chromosomes or so. And what you see I've circled here is where you see clustering of markers selected using the song method, and versus the bin method, because we force those markers into bins. And really the bin method is just using the song method, except forcing it to be broken up by bins, because we wanted that better distribution of markers than what you got with the song method. So overall, it gave us good polymorphism, gave us a good distribution, so we're pretty happy with that. So the next thing was to test the probe set in different germplasm and see how it performed. So we ran it with 96 soybean plant introductions, and we also ran it in this 84 rills from this Essex by Williams 82 population. This Essex by Williams population, I used it whenever I was at USDA. This population had been used to help assemble the soybean genome. So these rills had been ran with the 50K Infinium chip, which the 1K is a subset of that 50K. So we could compare concordance between the Illumina and the 1K MIP probe set. For SNP calling on the MIPS, we did trimming using Trimomatic. Mary Happ, PhD student in my lab, she developed the pipeline here. She used Trimomatic, Bowtie, SAM tools, and then GATK for the SNP calling and the post genotyping. So the performance of the 1K on the 96 PIs, 85% of the reads align to the target sequencing. So we're getting good enrichment for the sequencing for the 1K. The logarithmic, the 900 probe, the 1,000 probes, it's a logarithmic scale on the y-axis there. Overall, for this particular run, we had 245x coverage. 800 probes had greater than 10 reads. So about 200 probes are underperforming. The advantage of individual probe synthesis is you can increase the concentration of those low-performing probes and increase the number of reads you get for them. So we're going to be able to work on this probe set to increase the performance of it on that. We did have 53 probes that had over 1,000 reads, so those are working way more efficiently than what we would like. So we're going to work on either eliminating those probes or decreasing the concentration of those so that we get fewer reads, because they're taking reads away. Ultimately, we would like to get the distribution of these probes as flat as possible, or even distribution. The accuracy of genotyping, though, was 98% accuracy. So great accuracy. We were really happy with that. The thing about the PIs, though, is they're almost 100% homozygous. There's very little heterozygosity in these plant introductions. So to assess heterozygosity, we wanted to use this Essex by Williams. Even though they're rills, there's still 6% heterozygosity, and we wanted to assess that with this population. We ran it with this. Our depth wasn't as good as what we would like. There was about 44x depth, or about 45x depth on this particular population. 74 of the probes were to the wrong target, and so those are probes that we're going to be eliminating from the 1K. Just like with the PIs, 200 of them had low efficiency, so greater than 30% missing data. So we're either going to replace those with SNPs that are in high LD with it, or increase the concentration of the probe to see if we can't bring that missing data rate lower. 6 genotypes were dropouts, so just bad DNA extraction. So the final data set, we had 725 total markers, and 292 of those were polymorphic in this particular population. So about 40% polymorphism. Now, whenever we look at the SNP calling parameters, now this is a busy table, and this is my last table before I get to conclusions. But we looked at different parameters of the SNP calling because we wanted to see how that was affecting things. And so when we look at alignment sensitivity, the sensitive end-to-end had most of the effect. Well, what you can see, the sensitive end-to-end had most of an effect on missing data, a small effect, but it got missing data down to 6% or 7%. For mapping quality, what we see is the mapping quality of 30 did get our heterozygous. Our heterozygous accuracy, the average is 73% there, but the medium is 90%. So we got a pretty big difference between our medium and our average, and I think that's because of the X coverage being only 45X. Now, whenever we go to a read depth, and before that 6% missing data was a read depth of only 1, where we require a read depth of 5 or 10, then what you see is that missing data rate goes up to 8% or 13%, but it really didn't have a big effect on our heterozygous. We thought it would increase our heterozygous accuracy, but it's still at that 74, 76% and a medium of 90%. But to look at, and then the GATK confidence call. So if we use our final parameters that we're looking at, SNP calling parameters, if we use sensitive end-to-end, a mapping quality of 30, a read depth of 5, and a GATK confidence call of 20, our overall accuracy was 93% with a medium of 97. Our homozygous call, just like our elite PIs, was 98% accuracy, just like we saw with our PIs. Our heterozygous is 74%, and I think that's because of our low X coverage. So I think if heterozygous accuracy is important, then that X coverage needs to come up. And our missing data rate's around 8%, but then the medium is around 1%. So I think with a higher X coverage, as we look at that will decrease our missing data quite a bit. So overall, before I turn this over to Dr. Kaufman, our genotype, just some top 5 takeaways here. Genotype data does add information throughout the breeding process to help breeders make better selection decisions. Breeders do complain a lot about the weather, but there is actually a lot of science that goes into breeding. So before, none of my breeder friends talk to me again, they do a lot of science in developing new cultivars. MIP accuracy and missing data rate, what we found was 98% accuracy homozygous call, 74% heterozygous calls, and 8% missing data. Targeted sequencing with Mips can reduce the cost of genotyping lines to under $5 a sample. And currently, Leah McHale at The Ohio State University is the leading PI of a North Central research project where we're applying genomic selection in the public sector of soybeans. My lab is going to be genotyping about 12,000 soybean samples from different breeders to start applying this from 10 different soybean breeding programs. And so with that, I want to thank and acknowledge my collaborators, Aaron Lorenz, Vince Pantalone, Kijun Song, and Leah, and then our funding from Nebraska Soybean Board and from the North Central Soybean Research Program. So with that, Dr. Kaufman, I will turn it over to you.
Thank you, David. Thank you, Energene and Genome Web. In my portion of the webinar, I'll give a short intro to the company, its technology, and we'll illustrate the usefulness of SNP panels with one example of multi-purpose nested SNP panels in rice. Agriplex Genomics was formed in 2014. We are in Cleveland, Ohio, in the Bioenterprise building, just on the outskirts of Case Western Reserve University. Our Plexic procedure is an adaptation of a process that was developed originally by one of our founders, Dr. Alex Miron, for cancer screening. In the agricultural space, Plexic is a mid-density SNP genotyping platform that is filling the gap between single-plex reactions on the low end of the screening spectrum and chips and arrays on the high end. The Plexic workflow consists of crude DNA isolation. The DNA does not have to be of high quality to fit our reactions or our process. We don't really need a lot of quantity of it. Just a very minute amount is needed. The isolated DNA serves as the template for the primary PCR. And this is really where our value proposition lies. It starts prior to the thermocycling. It starts with PlexForm, which is our proprietary artificial intelligence software package. It is used to design primers, but more importantly, it is able to predict multiplexing options. So these designs enables us to simultaneously PCR few dozens of targets to few thousands in the same well or PCR reaction. The amplicons of the primary PCR become the templates of the secondary PCR, the barcoding PCR amplifications, where the amplicons each get unique tails that will enable their sequencing downstream. I'm sorry. Agriplex, a collection of 55,000 unique DNA sequencing indexes, allows the simultaneously sequencing of almost infinite or practically infinite large number of individuals. It is only limited by the sequencer's capacity. After this round of thermocycling is completed, we are pulling the different barcoded amplicons into just one container. And the pool is purified and quantified, and it's uploaded for sequencing on Illumina next generation sequencers. Now, the workflow has some significant attributes in way of efficiency, cost effectiveness, and adaptability. First off, the PlexCall workflow lends itself to automation. You can see in this slide that all the steps are set up by liquid handlers and are carried out on high throughput thermocyclers. So by that, it minimizes labor and the time and the cost. The workflow is flexible and adaptable. We're talking here about molecular breeding. I will throw into the pile also seed quality assurance. And both of those large applications typically require to genotype a large number of individuals. And not only that, they require the flexibility of adding or substituting some of the markers. The fact that Plexic panel is a collection of markers and not physically bound to a surface, like in chips, allows for SNPs to be added, omitted, or substituted, and that is to dynamically customize and alter the composition of the markers as you need them. Similarly, I mentioned before the number of individuals that can be tested is large and can be as large as you desire. The final step in this process is the analysis, which is done by another proprietary analysis, software package. So PlexCall, it enables to automate the analysis process. It gathers the data, or the data from the sequencer is being imported into it, and then it automatically makes the calls and can construct a concise report. So with this platform at hand, we partner with industry or academia or public sector to create SNP marker panels that will fit their various needs. We partner with the International Rice Research Institute, or IRI, and use their one rice custom amplicon, 1K rice custom amplicon panel to multiplex it, and this effort is actually our model for this kind of an effort, or this kind of a structuring or building crop-specific or life or species-specific SNP panels. The 1,000 SNPs, actually after a slight correction, it was reduced to 955. So the rice panel is a good example for a nested multi-purpose panel. Some of the SNPs here are uniquely designed to support one specific application, while others can contribute to just more than one analysis, as in for example, a marker that is useful in the genomic survey, or at the same time can be part of a smaller subset that will determine genetic purity or varietal ID. The RICA SNPs were selected to be used in molecular breeding, various molecular breeding applications, and as I mentioned, quality assurance applications. The bulk of the SNPs in this panel, 838 of them, are SNPs that are chosen to be, or because they are evenly distributed across the genome, and they were selected for genomic screening. Those markers are based on their robustness that manifests itself in a high call rate. They were selected on a call rate that is higher than 95%, and also on the minor allele frequency that is more than 0.4 or 40%, and that was determined by genotyping more than 1,100 IRI indica rice breeding lines and indica released varieties. In this slide, it shows the results of pairwise polymorphisms assertion or test between 87 elite indica lines. So the 85 of the pairs that were examined exhibited between 300 to 500 polymorphic markers, and the average number of polymorphic markers was 374. So I mentioned that the panel is a multi-purpose. The other parts of the panel include 96 trait-related markers. So those markers are linked to 58 different important genes or QTLs. And currently, Dr. Damian Platten of IRI and his collaborators are working on identifying yet another set of trait-related markers that we hope to incorporate into a multiplexed set that will be released as RICA version 4 of this panel. The panel also includes 22 SNPs that were chosen for their discriminatory ability amongst elite indica material, and they're used in the determination of genetic purity, varietal identity, and for hybridity. So those are more on the production quality assurance side of the spectrum. So overall, the nested multipurpose panel can be used for marker-assisted backcrossing, genomic selection, QTL studies, and QA applications as seed purity and genetic purity. In addition, there is a power of running such a panel, especially in a consortium setting. It creates a base or a common language to all the participants that enables combining lines from different sources when you know a priori which parents are polymorphic, for what traits or markers, and what are the genetic assets that they may contribute. Or in a company setting, it bridges the gap between breeding and supply. So new lines that are emerging from the breeding program are already screened for quality assurance applications. So in summary, what I hope you'll be taking home from this short presentation is the potential to satisfy efficiently varied genotyping needs by Plexi, the mid-density SNP genotyping lab platform, and by developing and utilizing a nested multipurpose SNP panel. And with that, I'll turn over to Dr. Ed Brookman from Energy.
Thank you, Benny. Before I get started, I'd like to thank both Benny and David for participating in this webinar today with me. I am the caboose here on a train which is running just a little bit long, so I will try to be brief. Let me take a moment and introduce Energene to you if you're not already familiar with the company. It is a young company that was founded in 2010. It is an Israeli-based company, and most of the employees are in Israel. I happen to be speaking to you from the US today. Energene is a biotechnology company that specializes in agricultural genomics. We provide computational products and services to R&D programs that are involved in crop improvement. And those organizations exist in industry, academia, and in the government. Energene itself does not have any lab capability. When I talk about genotyping or sequencing, we work with service providers. And I say that up front because it's often a question we get asked, and the talk that I'm giving here today is perhaps a little bit different than the two previous talks in that respect. Our employees have expertise in areas like bioinformatics, statistical genetics, computational biology, computer science, algorithm development, software development. So we really are a big data company, a big genomics data company, and we provide products and services across the spectrum from data acquisition to data management and processing and storage, analysis, interpretation, and visualization, all of genomic data. The goal here is crop improvement, and the means to get there today is often molecular breeding. Energene was really founded on the principle that a reference genome is a solid foundation for molecular breeding and most crop improvement programs in the 21st century. We offer products and services that address all levels of this hierarchy here, starting from the reference genome on the bottom left. Of course, I'm not going to talk about all of those today. What I do want to focus on is SNP genotyping and the diversity analysis that goes into it. So it's in the mid-range here of this hierarchy. In molecular breeding, we typically face a challenge, and that challenge is just simply put, the cost of genotyping. Genomic selection is a great technique. It can accelerate plant improvement programs. It can create new expectations about progress that are expected from plant improvement programs and breeding programs. And it may be difficult in a breeding program to actually achieve those expectations today through conventional means. And so in many cases, molecular breeding is a necessity, and the cost of genotyping is simply a reality that has to be addressed. Genomic selection is a particular form of molecular breeding, but one that has come up a couple of times here, and that's what provides the background for my talk today. Genomic selection can require dense genotyping of many samples, and the cost of genotyping can be quite high. So what I would like to talk about today is to sketch out or outline some of the things that are possible with appropriate data analysis and data management that can help reduce the costs of genotyping in a molecular breeding program that employs genomic selection. Basically, the solution involves genotyping fewer SNP loci. Accurate predictions in genomic selection rarely require more than 5,000 or 6,000 optimized SNP loci, and the number can be much lower than that, as we've already heard earlier. But we can go further than that and point out that direct genotyping of a much smaller number of loci And the imputation of data for ungenotyped loci is feasible and is extremely feasible and is a way to save costs. So the basic message here is to genotype less and impute more data. And these factors here are especially true for plant improvement programs and breeding programs that have highly structured populations in which a lot of linkage disequilibrium exists. So this is cases where we can really drive down the costs by reducing the numbers of loci that are genotyped and doing very accurate imputations from those loci to a larger number of SNP loci. So the way this works in detail follows a couple of different steps. The first thing that you have to understand is the linkage disequilibrium that exists in your breeding germplasm amongst the SNP loci. And the fastest way to get to that today is by doing some sequencing in your breeding germplasm and analysis of that sequence data to determine the linkage disequilibrium and the haplotype structure of the germplasm. Armed with that information, it's possible to optimize SNP loci within the germplasm that will provide a sufficient number to discriminate the haplotypes that exist in your breeding populations and support an application like genomic selection. Another step of optimization is to subset even further to identify a much smaller set of SNP loci that are suitable for direct genotyping. So these are the loci that will be genotyped in the lab, and then a larger set of loci will be imputed from those data. There has to be an imputation pipeline in place, and it simply takes the data from the genotyped loci and imputes data for the ungenotyped loci. And of course, it's important after all of this to select an appropriate cost-effective technology platform and a high-quality service provider. And we've seen some examples of both of those actually in the previous talks. So that's the basic sequence of steps that has to be followed. This slide is just a schematic illustration of what I described on the previous slide. On the left, we have that initial sequence analysis and SNP optimization, which in between looks at the linkage disequilibrium and the haplotypes that are present in the germplasm. On the right is where the real gains take place, and that's where we genotype low-density data and impute high-density data from that low-density data. Imputation, actually to some people, sounds like just making data up out of thin air. It's not true, although I understand why it sometimes sounds that way. Imputation has a long history, and it's been well used in human genetics and in animal genetics. It is coming to plant genetics relatively slowly, however. This is just a collection of headlines from the scientific literature over the last 20 years. Imputation is not something that is unique to Energene or anybody else. There's a lot of activity in this area, but it is something that Energene has invested heavily in so that we can assist research organizations in reducing their genotyping costs and taking full advantage of the best in imputation algorithms. This is just one example of what can be achieved with imputation, and the focus here is on the accuracy of imputation, which is often a question that we hear about. So for this experiment, we have 92 DH samples from maize that were genotyped at a large number of loci. And from those loci, we can select subsets of loci and treat those as the genotyped loci, and from those impute the ungenotyped loci, which are simply loci that were genotyped, but we're masking those data. On the horizontal axis, you see the number of loci that were included in the subset that we're calling genotyped data. And we have two vertical axes here, one on the left, which is the number of imputed SNP loci from the genotyped loci, which is represented by the blue bars. And perhaps more importantly for most people is the yellow line, which represents the accuracy, here presented as the error frequency of the imputed loci. And you can see that with a very small number of genotyped loci, the accuracy is lower, or the error frequency is higher, close to 1%. But as we increase the number of genotyped loci within the population, we can impute more SNP loci, and at the same time, the error frequency of that data goes down to a very small number. This is a little bit misleading in the sense that one might think that we have to be out here on the right side of this graph for breeding purposes, but that's not true. In the case of breeding populations for crop improvement, the right half of this diagram is irrelevant. We'll typically be operating on the left half or even on the left third of this graph. We really want to be genotyping a number of loci that is in the range of 500 to 1,000 at most. And the error frequencies that are involved in those ranges is not high and is completely acceptable in the context of a technique like genomic selection. So finally, I'd like to finish up and just summarize some of the value that this has for breeding programs. Above all, we want to reduce the cost of genotyping and make genotyping feasible for conventional programs as they convert, and to make it as accessible as possible for all breeding programs in plants. So the way that we do that is by minimizing the number of SNP loci that are genotyped in a laboratory situation, which is expensive, and maximize the number of SNP loci that are accurately imputed. Another factor here, which is important, is that when we think about genotypic data and our allocation of limited resources in a breeding program, we gain a lot of flexibility in where resources can be applied. So one place they can be applied is in sequencing the breeding germplasm, and that sequence data on breeding germplasm is permanent and can be leveraged for many different purposes, as opposed to the genotypic data on breeding populations, which is basically just used once and is mostly discarded as selection decisions are made. Although I don't point it out in this slide, it's also true that if you can reduce the cost of genotyping by reducing the number of loci that you genotype, you can effectively expand your breeding programs by genotyping more lines within your breeding populations with some of your savings. The final thing I'd like to focus on here is just the goal in genomic selection, which is not the accuracy of imputations, but is the accuracy of predictions. Imputation accuracy or genotypic accuracy on breeding populations has a limited impact on prediction accuracy compared to some other factors. And it is well to, again, consider a resource allocation in this context. The prediction accuracy in genomic selection is much more influenced by things like the quality of the phenotypic data that's collected for the training sets, the relationship of the training environments to the testing environments, and the genetic relationship of the training set to the breeding populations. The accuracy of genotypic data on the breeding populations really has a small influence, and frankly is one of the easiest ones to address and measure in a situation where we have the appropriate things in place. So with that, I will turn it over to Christy for a few minutes of questions and answers. Thank you.
Great. Thank you all very much. As a reminder to webinar participants, if you have a question, you can type it into the Q&A panel at the bottom of your screen. Which appears on the right side of your screen. Before we begin the Q&A session, we'd like to ask all of our participants to stick around at the end of the webinar and fill out our exit survey to let us know how we did. All right, so let's start our Q&A. So David, our first question is for you. Why did you choose for Mips to do genotyping instead of multiplex PCR type of approach, such as AmpliSeq, AgriSeq, or AgriPlex?
So with part of our goal was we were really looking for something that we could run in-house, mainly because we're trying to push the cost down as low as possible. So, AmpliSeq, AgriSeq, those were, there was TruSeq, AmpliSeq. Those were certainly, at the time where we started this about 5 years ago, those were viable options, and they are very viable options. But it was something, there was lots of literature available on Mips, and it was something that we could run in-house without the need of buying kits. So it was mainly, I saw it as an option for us to try to drive cost as low as possible. Yeah. Christy, were you able to hear me there?
Is somebody muted, maybe?
Ed, could you hear me?
I could hear you.
Okay.
I can hear you too.
I think we lost Christy.
All right, I'm going to jump in here. This is Allison from Genome. Let's move on to the next question. Is it possible to use and adapt the AgriPlex approach to measure DNA methylation by, for example, first doing bisulfate conversion followed by your targeted sequencing approach?
I'm not sure that I know the answer for that because I have very little, actually I have no experience with DNA methylation. But I would assume that you can do some sort of a subtraction analysis that is to do the regular PCR and then use it on samples that you'll somehow, the methylation prevent some of the amplification and subtract one from the other. But I'm not sure if I'm answering your question here or just showing my ignorance.
That's okay. I'll move on to the next question.
Allison, if you like, I can take back over. Sorry about that. I had an internet blip, as so many of us do these days. David, I guess we can go back to you. Can you tell us how much Mips can reduce costs?
So that's something that we're still trying to drive the cost down. Like I said, we're below $5 a sample. Our goal is to get it down to $3 a sample, but it's still a matter of getting that DNA extraction is that $5 doesn't include DNA extraction. So I think that's right now our biggest mountain right now is trying to get a cheap DNA extraction that's clean enough for it to work with. But I think we can get our volumes down low enough I think we'll get closer to 3 than we are to 5 right now.
Great, thanks. Ed, a question for you. Could you tell us the criteria for accurate imputing?
So basically that is a error frequency. When we have observations on loci that we're going to impute, we impute those loci and we compare them to the observations that we had previously obtained. And so we can compare the imputed data to the observed data, identify those observations that are concordant and those that are discordant, and the frequency of discordance is the error frequency in the data.
Great, thanks. And David and Benjamin, sorry, I'm just finding my place. Oh, David, can Mips be genotyped on Thermo Fisher Scientific's Ion 5? And then Benjamin, following up to that, the same question for Plex-Seq.
So, I mean, I have not tried it, but I would expect if you could, I would think if you could replace with the universal PCR, put the right adapters on so that it could go onto an Ion, I would think you would be able to do it, but I don't know for sure about that.
And Benny?
So I guess part of Dave's answer is good for us as well, because we're using those Illumina indexes and tails that are essential for the sequencing. The other part of it is the fact that the software that is used for analysis will have to go through some major adaptation to be able to be used on other sequencers. But I want to add, and really this goes also for the first question about the methylation. I will be glad to discuss this offline, because with somebody on the methylation side that can fill in my gaps, and we can have a fruitful exchange this way.
Great. I'm afraid that's all the time we have for today. We apologize for the short Q&A session, but we have run out of time. So we'd like to thank our presenters and our sponsor, Inversion. If we didn't have time to get to your question, we will follow up with our experts. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.